Hi everyone, I'm Dominique Makowski. I'm a lecturer at Sussex, and today I will talk to you about the links between physiology and cognition, and more specifically, how we can I have no financial interest, but I need to disclose that to give you the outline of this presentation. First, I'm going to make some general points about some general distinctions about reactive and predictive physiology. And then I will essentially go through some of the most popular physiological signals, such as electrodermal activity, respiration, cardiac activity. So I'll start first with a somewhat provocative statement, which is physiology neuroimaging, or rather, could physiology be, included in the different neuroimaging techniques? And at first glance, it might be seen as a bit awkward because neuroimaging has to do with the brain and physiology has mostly to do with the body. However, I suppose one of the important piece of this question is how do we define neuroimaging? And I would like to first stress and emphasize that even though there's a fairly strong bias in favor of that, especially at OHBM, but I would like to mention that neuroimaging is not a synonym for the MRI techniques. And I think that, part of the kind of equivalence that could have been sometimes made is that the word images from neuroimaging has been maybe taken literally as, the image of a brain. And it's true that most of the visual representation of MRI gives this very realistic image of the brain, even though we know that it's a deep and complex reconstruction process. Nonetheless, it gives this impression. But neuroimaging really needs, the word image has to be taken in a broader sense as just a representation of brain activity. For instance, EEG, which arguably belongs to neuroimaging. Most of the ways that we represent EEG signals, whether it's different waves, different time frequency kind of plots, most of them don't look like brains, even though we can also do that, and nonetheless it is part of neuroimaging. So if we define neuroimaging generally as a measure of objective brain-related activity, fMRI, for instance, measures the Bolt signal, whether it's electric signals or glucose activation, or all kinds of different brain-related activity. Then we have to wonder whether physiology, kind of qualifies with this definition. And we know, we have first to acknowledge that there is, we know that there is a very strong interaction between the body and the brain. And most importantly, the body often seems to reflect neural mechanisms. And what I mean by that is that typically we could argue for this kind of causal pathway in which, when, for instance, when we feel stressed, this is related to some kind of activation of a particular pattern of brain networks. And this is again related to some. And could thus be seen as belonging to the realm of neuroimaging. But it's not that simple, actually, and I wouldn't be necessarily keen on including physiology as, part of this big neuroimaging realm. Let's take, for instance, one physiological signal, which is the cardiac activity as measured by the heart rate. Obviously, this heart rate is, the heart rate activity is partially triggered by signals coming from the brain, which can eventually, through different systems, can accelerate it or decelerate this heart rate. So there are top-down influences. Rapid changes are typically under the control of the autonomic nervous system, which is kind of an adaptive system related to or influenced by cognition. And there are also slow changes that are happening for the heart rate that are typically related to background processes such as circadian rhythms, sleep cycles, metabolism, changes in temperature, and hormonal systems. So if we considered only this, then fair enough, it is reflecting some changes happening centrally in the brain. However, one of the key things is that there are also very strong bottom-up influences in that most cognitive processes actually take into account or use information coming from the body, the bodily state, to run essentially. And what And I would like to start this presentation with an important distinction, even though it is fairly artificial in the sense that in reality, the different indices are, somewhat intertwined and related. Nonetheless, I think it's very useful to kind of understand what we can do with physiology in terms of, how to include that in your experiments, how to interpret these different indices. It's very useful to have this structure and this distinction in mind. And the distinction is between reactive and predictive physiological features. And by features, I mean anything that can be really quantified or extracted from physiological data. This can include the change of heart rate, change in the amplitude of some signals such as skin conductance, the current respiration phase, whether it's inhalation or exhalation, and most of these features that can be extracted even at one moment in time or over an interval could be separated into those that are reactive and the ones that are predictive. In other words, that are top down and bottom up. Or another way to put it is to think in terms of a kind of simple regression model where you have a dependent variable predicted by an independent variable. So reactive features or the reactive indices will mostly take the place of a dependent variable. It will be your outcome variable. It will be, you will have as a predictor, for instance, I don't know, maybe emotions or the attentional engagement, and this will cause some changes in this reactive physiological indices, for instance, a higher heart rate. And for the predictive features, it will be the opposite, meaning that predictive features will most of the time be considered as a predictor in your model, as an independent variable. This is the case, for instance, with heart rate variability indices or the respiration phase, whether you are, when something appears on the screen, when the stimulus appears, or when the participants do a response, whether the respiration phase is inhalation or exhalation might have an impact on the timing of this response or something else. So these would be mostly predictive of cognitive outcomes or behavioral outcomes. And I would like to draw another distinction within these, in particular within these reactive features. They are the ones that can be considered or can be used in event-related paradigms, and the ones that would be seen as state-related. And the difference between them is that most of the time the event-related features is something that we can quantify directly after an event. So typically this, in event-related paradigms, you show a stimulus or something happens, and within a couple of seconds after, you measure the changes. With neuroimaging, it will be in the brain, but with physiology, it will be you measure the changes in physiology, in the bodily response. So typically, the short-term heart rate changes, or the respiration rate changes, or the amplitude of the respiration that follows an event can be considered to be event-related physiological indices. And another kind are the state-related, and these are mostly, for fMRI folks, it will be akin to kind of when you have block designs, when you have a longer kind of time interval, a longer time period of several minutes, you can then compute some sort of physiological index to quantify and to characterize this kind of longer state, which can be then used to correlate with this condition or this state or this population, for instance. So this would include indices such as indices of heart rate variability, or the variability of respiration rate, or the number of skin conductance responses that happen over a period of time. For instance, if you have two conditions in your experiment, you have participants watch a relaxing movie versus an emotional movie, and you have these two emotional versus neutral kind of condition, you could measure the number of skin conductance responses in each of these conditions, and investigate whether there's a significant difference between them two. So some of these measures can also be sometimes used in a predictive fashion. Nonetheless, predictive features will most of the time include things like the momentarily heart rate. So this is like, for instance, the heart rate when something happens on stimulus onset. So we know that if you have an elevated heart rate, this will influence the way you perceive or you pay attention to things. The cardiac phase also I will come back into that in this presentation, but depending on the timing of an event within your cardiac cycle, it might lead to different behavioral or cognitive outcomes. Same with respiration rate, respiration phase, and so on. So now that we are a bit clearer on the different types of indices, let's start with the first kind of signal that we often encounter in physiological research, which is electrodermal activity, also referred to as EDA. So you will see electrodermal activity referred to under different names, or rather different names are more or less related to electrodermal activity, such as skin conductance response or galvanic skin response, GSR and SCR. As we will see in a second, these are not necessarily entirely equivalent, so there are some important distinctions between them. So electrodermal activity is measured via the current difference of conductance between two electrodes. So it's most of the time measured on the fingers with two electrodes wrapped around the tip of the fingers, but it can also be attached to feet. And it quantifies the conductance of the skin, which is influenced by the activity of sweat glands itself, related to the autonomous nervous system activity. In other words, the more the sweat glands activate and the more the skin is sweaty and the more this conductance increases. And it's a fairly cheap and easy thing to measure, and it's also quite straightforward to analyze with some important considerations. So EDA is actually a combination of two distinct components, which we refer to as the tonic and the phasic components. As you can see on this figure, the raw data is the top signal on the top, and you can see that it's made of this kind of slow pace signal, which is the tonic component, and a more kind of flat with peaks signal, which is the phasic component. The tonic component is also referred to as the skin conductance level, SCL, and it corresponds to slow changes driven by background processes such as thermoregulation and general arousal. And the phasing component is typically what we refer to when we write skin conductance response, because these are fast changes that reflect the sympathetic autonomic activity that is typically associated with emotion or attentional processes. So these are really the kind of phasic response to some cognitive process. And so one of the key steps of EDA analysis is to do the signal decomposition, because what you have when you record the signal is the raw EDA, and then you have to apply some algorithm. It can be just a filtering to then extract the tonic component and, most importantly, the phasing component. And most features in psychophysiology, meaning that most in the realm of cognition and neuroscience, we are mostly interested in the phasic component of EDA. And this phasic component includes peaks, which are the skin conductance responses per se. So they have this very kind of normal shape in a way, distinguishable shape. The only one of the limitation, or one of the things to take into account when using EDA, is that it has a fairly slow dynamics, meaning that this kind of skin conductance response unfolds over a couple of seconds, somewhere between 5 and 10 seconds, and then takes some time to return to a baseline. And so this is not necessarily suited for fast-paced event-related paradigms, where you might have some convolution between the responses. And so from these peaks, we can essentially extract different features. Some of them can be considered as state-related features. This would be typically the number of peaks in a given interval or condition or task or population. So again, the example is that if you have one interval where people are anxious and one interval where people are relaxed or neutral, you would simply count, you would decompose the signal and then count the number of peaks in each condition. And this number would give you your dependent variable. But it can be also used as an event-related feature, meaning that in this case, what we would typically do is that we would consider after each stimulus, after each event, we could do an analysis on whether it is followed or not by this skin conductance response, or we could also analyze some of the characteristics of this response, such as typically the amplitude, which is the difference between the peak height and the height at the onset of the response. And what you can use electrodermal activity for, you can use it. It can be useful to have as a manipulation check. For instance, if you have a paradigm that has different tasks or conditions that are supposed to elicit affective responses, typically in the field of affective neuroscience, then it would be very useful to have electrodermal activity here to also show that whatever you observe in the brain and even behavioral correlates, you also show that this condition or this task indeed elicited some typically affective responses. But it can also be used to eventually give some insights on whether this task was difficult or effortful, because this skin conductance response is not very specific. It's sensitive but not necessarily specific, meaning that this response is the same. It's most of the time, it can be for affective reasons, but it is also sensitive to the attentional engagement or salience detection, essentially. And it can provide additional evidence. So in a paradigm that is interested in emotion, in the emotional experience or the emotional regulation, it can be very useful to know, for instance, what networks or what brain regions are activated and the strength of this activation in relationship with some experience or regulation technique. But it's also interesting to know whether this activation is correlated with some changes in the bodily response. And similarly, it can be also useful in instance research that is investigating, salience or detection of certain events to be used as a marker of bodily reaction. Let's move on to respiration. Respiration is traditionally, or at least the easiest way to measure respiration is with a respiration belt that is strapped to the chest, and the expansion and contraction of the chest during breathing creates this oscillatory signal. And the signal contains some important features, which are the peaks and the troughs. And these peaks and troughs allow us to determine the respiration phase, which is either the inhalation, which is when the signal goes up, followed by an exhalation when the signal goes down. So this peaks and troughs identification is the key part of the respiration signal processing. And the respiration phase that I just mentioned can often, has been used as a predictive feature, meaning that there are some studies showing that when something happens, when a stimulus occurs, depending on whether it occurs during an inhalation phase or an exhalation phase, this leads to different behavioral outcomes or also different brain activity patterns. Another thing that we can analyze is the respiration rate. So that would be the duration between each peak of the signal, as well as its variability. So typically this would be used as a state-related feature. If we take our example of having two conditions. One condition where participants maybe are stressed, or they did some, or it just follows some physical activity, versus another condition where participants are in a relaxed and neutral state. Then we could compare the breathing rate between these two conditions. And finally, technically, we could also look at what happens for a respiration cycle following a certain event, so in an event-related fashion. And we could look at characteristics of this respiration cycle, such as the amplitude, which would be the height of the peak, as well as other characteristics in the respiration phase asymmetry. So as you can see, the inhalation is typically shorter than the exhalation, but this ratio could eventually potentially change depending on some circumstances. However, I must say that there is paradoxically little research on respiration, especially on its direct links with cognition or neuroscience. And this is a bit odd, given that respiration is one of the primarily target for different interventions, including meditation, relaxation activities, or sports science. And I believe one of the reasons for this scarce research is that the dynamics of the respirations are fairly slow. Each respiration cycle takes a couple of seconds, and it's also fairly noisy, meaning that the changes that we might observe are not necessarily highly robust. So nonetheless, there are a few studies investigating that. For instance, there is this paper showing a correlation between trait anxiety and the respiration rate at rest. And there are also other studies suggesting some impact on the reaction time or on the activity of different brain networks. And now let's move on to the final signal that we will cover today, which is the cardiac activity. So probably one of the most inexpensive and easy way to obtain cardiac data recordings is by measuring the pulse. This is typically done by the photopletismogram, more often referred to as PPG, which is nowadays implemented in really a large number of wearables. But recently there have been some developments to even do that remotely. So apparently, if you filter correctly data from a commercial webcam, you can actually, through the color of the skin, you can derive a PPG-like measure. It has some challenges, but there are some studies doing that. Nonetheless, so the way it works is it sends some infrared light, and it has a photosensor on the other side, and it measures the peripheral blood flow. So every time the blood flow reaches the arteries, there is the capillaries expand, and this changes the amount of color that essentially passes and reaches the photosensor. But something important is that this is only an indirect measure of cardiac activity. It doesn't measure cardiac activity per se. It measures rather the blood flow related to each cardiac beats, which means that it's also typically a bit less precise than ECG because there's a delay, and there's also some, depending on if you record it from the fingers, for instance, depending on whether people have longer or shorter arms, it can vary the delay between, say, a heartbeat and the time it reaches the the veins and the arteries in the hands. So another way of measuring cardiac activity that is more, more accurate, I would say, is via ECG. So this is done for medical purposes via a multi-lead ECG. So there are usually a certain number of electrodes, 6 electrodes that are placed on the chest. And these 6 electrodes, essentially, they triangulate the brain in from different angles. And this gives you different types or different signals or different ways of looking at the cardiac activity. And so from there, we can diagnose certain pathologies, because some of the features of a cardiac cycle are more prominent on some leads as compared to other leads. However, in research, and especially in psychological research or neuroscience, we more often use a single lead ECG. So it's only recorded with 3 electrodes that are, for instance, that can be placed like this in a so-called lead 1 position, or I would recommend placing them like this, which is referred to as a modified lead 2 position, and has the benefit of it enhances the R peak, which is one of the main features that we aim at detecting in ECG signals. So again, the idea is that depending on how we cross the heart, depending on the angle, this will change the shape of this cardiac signal. And so many sometimes noisy signal can be acquired because of this electrode position is not optimal, or it's just sometimes wrong. Sometimes we connect the wrong electrodes and it just inverses the signal, for instance. So this is how an ECG look like. So it has these fairly seemingly regular, at least, rhythm of very high frequency peaks, very sharp peaks, which are the R peaks. So this is one of the most prominent feature of an ECG signal. This is what this is the first kind of, when we pre-process ECG data, this is the first thing that we look for, because once we have the R peak, then we can eventually try, if the signal is clean enough, we can eventually try then to delineate the cycle and find the other different waves. And so each of these different waves in a so-called QRS complex, which is the name of a cardiac cycle, refer to different mechanisms happening, different motor mechanisms happening during the heartbeat. And so the ECG is really a direct measure of the electrical cardiac activity. So something similarly to the respiration signal, I mentioned that there are 2 phases. There is the inhalation and exhalation. There are also the cardiac cycle has been traditionally dissociated into 2 discrete phases, and these are the systole and the diastole. And one of the way of classifying them is that from the R peak until the end of the last kind of cycle, until the end of the T wave, this is the systole phase, and this is where the blood is ejected from the heart and passes through the arteries, and this is where the baroreceptor, so the pressure sensitive receptors that are in the arteries, will get activated and will fire maximally, and then it kind of and it's believed that there is a sensory attenuation happening during systole caused by this baroreceptor activation. And as a matter of fact, there are some studies investigating different consequences, I would say, cognitive consequences of this cardiac phase. So for instance, it has been shown that it can modulate this activity of the baroreceptor that projects on certain brain areas, can modulate things like nociception, emotional processing, certain features of cognitive control such as conflict monitoring maybe, or inhibition, time perception, as well as the activation or frontal areas. And so we could use that as a predictive feature, meaning that again, for instance, we have some research that when we look at the cardiac phase on stimulus onset, and we show that this, depending on whether the stimulus appears during systole or diastole, this kind of, modulates the participants' behavior. They would be faster or have poorer inhibition skills during systole, for instance. And another key characteristic of cardiac activity is the heart rate. And in order to compute heart rate, we need to get the location of the peaks, the accurate location of the peaks. And so this can be derived either from ECG, again, the most precise, or from PPG as well. As you can see on the figure, the two are slightly shifted, but for most case and purposes, the reliability from a PPG is more than enough, especially to compute things like heart rate variability or the mean heart rate. And so once we have the location of the peaks, we then compute the interval between them, which is called the RRI, the interval between each R peaks, which is our discrete heart period, and then we interpolate the signal, and this gives us then we convert that into heart rate. You can see in this figure here below that the heart rate is low when the R peaks are close to one another, and the heart rate increases as the R peaks are more further from one another. And so the heart rate is often used as a state-related feature. So what we do is we compute the average heart rate over a given amount of time. It can be, a few seconds, a few minutes, a few hours, and the average heart rate is associated with a number of things. The most basic one being physical activity. So if you do some physical activity, your heart rate increases, and same goes as well for affective tasks or affective conditions. However, what we can also do is to take the heart rate at stimulus onset and use that as a predictive feature. If you experience an event or a stimulus and your heart rate is already high, then this likely changes the way you process this event, and vice versa. If you experience an event with a very low and relaxed heart rates, the experience and the processing is also likely affected. And the heart rate also, beyond just the heart rate or the mean heart rate, the changes in heart rate are particularly important. And something that has been traditionally looked at is the short rate changes within a window of about 5 seconds. And what we have observed is that in many cases, when something happens, whether it's an emotional event or something that captures our attention, there is a momentarily heart rate deceleration. As you can see here, the stimulus happens at 0, and the heart rate kind of decreases within this about 5 seconds window. And this has been linked with affective processing, as well as also attentional orientation and engagement. So it seems to be not only emotions, it might be something a bit more general, just as attentional orientation or salience detection, for instance. And so this is traditionally used as an event-related feature, meaning that for each stimulus, we would, for each time-locked event, we would look at at the amplitude of this deceleration, for instance, by computing the minimum of the heart rate minus the baseline. And we would have this value for each different stimulus, and then we would do some within participant kind of analysis. And importantly, this is a event-related form of what is known as heart rate variability, because typically this is just a change in heart rate. But when we refer to heart rate variability, usually we mean something a bit different. And we typically mean a state-related feature that can be computed, for instance, from a resting state paradigm. But it can also be computed from even longer timescales. So in some cases, people are connected to an ECG or a PPG throughout the entire day, and we measure the heart rate variability over several days, or hours, or minutes. So there is some debate about what's the minimum kind of window necessary to reliably capture heart rate variability indices, and it most likely, it depends on what indices we use. It's one of the most popular physiological index, meaning that it has been really used a lot in a wide range of cases in literature. The general idea is that the higher the heart rate variability and the better the outcomes are. And the assumption is that if we have a high heart rate variability, a very kind of non-predictable one, very chaotic, very pseudo-random, it generally indicates that this heart would be adaptive, meaning that it adapts to different events, whether it's external events or internal events. When we think of things or when we experience different things, and this simply means that our body reacts, according to what we experience, and it's correlated with a number of positive outcomes, whether it's physical outcomes, like physical health, as well as mental health. It's fairly predictive, or at least correlated with a number of disorders. But the problem is that heart rate variability alone as a term doesn't mean a lot. There are essentially more than 100 different indices that can be used to quantify heart rate variability. And so I'm not going to enter into the details of each metrics, but simply maybe to let you know that it is not something that is complicated to compute. So for instance, in the packaged neurokits, there is a function called HRV that you can just run on a signal, or you just provide an ECG or a signal, and it will compute almost pretty much all the indices that are traditionally used in the literature. So them is not really the issue. The problem lies with how to interpret them, and how to not cherry pick, and how to what to report, and so on. So this is an active field of development, actually, the kind of standardization, interpretation, and even the understanding of these different indices. So if there is a couple of tutorials, we've written some about HRV indices, if that's something of interest, how we can classify them and how we can compute them. We've done also recently we are doing a bit of work to try to objectively cluster them together. And it seems like, all these different indices can be grouped and do do refer to various aspects of this of such variability. For instance, the kind of centrality or the average heart rate, the dispersion, the frequency, the dominant frequencies, the complexity, the harmony of the rate, and aspects like that. But this is still very much work in progress. And so what you can use cardiac signals for? Well, it's very popular and to correlate resting state HRV. So if you have, a resting state kind of paradigm, you can correlate the HRV with a number of things, such as brain activity patterns. You can compare different populations, correlate that with cognitive abilities as well. And yeah, it can cast light on the bodily state of your participant. And yeah, it's a very it's a well-tested and fairly robust approach to explore and investigate the tight coupling between the body and the brain. So, in conclusion, I have made a little decision chart. So if you already acquire physiological data as part of your neuroimaging experiment, well, then you could use it to strengthen your conclusions and your claims. Essentially, you can use this data to corroborate your findings if you, assume and conclude to the existence of some cognitive process. You can see whether this is in line with the bodily reactions or maybe, or not. So it can be used as essentially additional evidence. And also it can facilitate, data interpretation in some cases where the findings that, are unclear. And the second thing is that I would also suggest you to make this data available, even if you don't use it directly. So you can do so, you can put it on Open Neuro, thanks to some initiatives like Phys2Bits. But even if you don't use this data as part of your, main experiment or paper, it can still be useful to other researchers to either test new hypotheses, or maybe even to students, that can use this data as part of a secondary kind of data analysis project. It's a very good introduction to signal processing and to neurophysiology, and so it can have some educational value as well as research value for other researchers. And if you don't yet acquire physiological data, well, I would really I hope I have convinced you a bit or have given you some ideas about use that in your research. And I would really emphasize the kind of value for money that physiological research has. So the recording equipment is nowadays, very cheap. For instance, we use Betalino kits that are sold for a couple of €100, and that are able to record simultaneously all the most important signals. So it will be a worthwhile investment. And yeah, maybe it would allow you to also include, it can also help you to do and to run pilot studies. So running fMRI experiments is very expensive and costly, and so before that you need to run pilots, and maybe it could be good you could use physiological data in your pilot studies to have a bit of objective measures as well, and to essentially build better pilots and be better prepared and have better hypothesis when it comes to your final kind of big neuroimaging experiment. So yeah, with all that, I thank you very much for listening, and yeah, if you have any questions on physiology and how you can use that in your research, don't hesitate to to get in touch with me. And yeah, thanks to the organizers of this symposium. See you.